Thank you for giving me this opportunity to come to this meeting. And today I'm going to talk about hijacking of the CREB into nucleus by FKBP51 is central to the control of innate immune and anxiety. Anxiety disorder is a common disease that is with a incidence rate about 7.3 worldwide. And it concurs with many other diseases and make this mechanism a little bit complicated. And so far, people have several hypotheses on the molecular mechanism of this disease, including neurotransmitter hypothesis, which suggests that, all the transmitters, serotonin, GABA, might be involved in it. And neuroendocrine hypothesis, suggesting the HPA axis may be involved, and neuroimmune hypothesis, which is supported by a lot of data that those inflammatory factors are consistently high in those patients. And the treatment of anxiety disorder so far, we have SSRI, we have benzodiazepine and other behavioral therapy. However, that people are still looking for fast-acting drugs and more drugs with different variations to handle this disease. The molecule we're interested in is FKBP51. And what is very amazing about this molecule is a very conserved family of a chaperone protein. This chaperone protein, it has a peptidyl prolyl trans isomerase, PPIase. Why is important for this is because this enzyme can change the conformation of proteins and make them active. And this is the 3D structure of this molecule. It has FK1, FT2, and also TPR domains that interacts with different molecules. And we're particularly interested in its regulation to activate the transcriptional factors. Previous study has shown that, FKBP51 promotes I-kappa B alpha, and then to modulate NF-kappa B, which is also very important for inflammation and psychiatric disorders. Also, FKBP51 binds AKT and promotes the dephosphorylation and cause change of growth, proliferation, survival, etc. FKBP51 also inhibit the transcriptional activation of GR, MR, and PR, those nuclear receptors, and it's known it can promote the transcriptional activation of AR and ER. So that we like this molecule because it is a very important molecule, particularly for transcriptional regulation. And it has been shown FKBP51 is associated to stress, that its SNPs are associated to development of stress-related disease like depression, PTSD, anxiety disorder, and aggressive disorders, aggressive behaviors. And also long-term cortisol treatment can induce the demyelination of FKBP5 genes, which will cause the sensitivity to stress event. Also, stress stimulation and HPA axis hyperfunction also can increase FKBP51 expression, which may be involved in the development of depression and anxiety. However, whether FKBP51 can modulate CREB is still not unknown. CREB, everybody knows it's a very common gene that it's a CREB response element binding proteins is known to associate with stress and anxiety. CREB is important because it modulates the transcriptional regulation of many molecules involved in mechanism, transcriptional regulation, cell growth, and cell survival. And there are many studies has shown that CREB is involved in stress-related disease, in acute stress or chronic stress, or high cortisol levels. The activity and the expression of CREB in amygdala and hippocampus has been changed. It may be involved in the promoting anxiety-like behavior. And also stress can promote in CREB activation in nuclear compounds. That may also cause the increase of anxiety-like behavior in animals. The SNPs of CREB genes also increase the risk of stress-related disease. There are still many differences showing, many previous studies showing that CREB may be involved in the stress-related diseases. However, FKBP51, how FKBP51 regulates CREB is still not being exposed. Therefore, we have this hypothesis that the stress may leads to the activation of CREB, then modulate the neuroimmune system. Stress, this mechanism may be involved in the anxiety disorders. So that CREB is a key molecule, and can FKBP51 regulates CREB, then cause this neuroimmune regulation, then finally it will affect the anxiety disorder. This is our hypothesis. So first of all, we want to see if corticosteroid regulates the formation of FKBP51 CREB protein complex or not. So we used a neuronal culture and treated the cell with a quart and different concentration of quart, 100 nanomolar and 1 micromolar, and trying to see if the co-immunoprecipitation of CREB and the FKBP51 is increased or not. And we found indeed there is an increase of the complex formation after 30 minutes of treatment with a quart. To confirm this, we used the double immunostaining, and we found that after 30 minutes of quart treatment, there is a significant increase of FKBP51 to the and the co-localization of CREB and FKBP51. You can see that the yellow dots are co-localizations. And after 24 hours CREB treatment, we still can see this increase of co-localization. And after 3 days treatment, which is mimicking the chronic stress process, you can see that in 1 micromolar CREB treatment, the co-localization of FKBP51 and CREB becomes very weak. It's after 3 days of treatment, this co-localization is significantly reduced, which is suggesting during the chronic stress, this mechanism, this FKBP51 and the CREB interaction may become much weak. Next, we want to look at whether corticosteroid regulates the expression level of FKBP51 and CREB proteins in the nucleus. Because that CREB is a transcriptional regulator, so it's functional when it get into the nucleus. We also do a after hippocampal neurons were cultured for like 12 days, and then we treat these neurons with CREB for 30 minutes with various concentration, 100 nanomolar and 1 micromolar. And you can see that the co-localization of FKBP51 with the nuclear dye is quite is increased, and so does the did the CREB. The CREB is also the CREB and the DABI co-localization is also increased. That suggesting that the CREB get into the nuclei. After 24 hours of CREB treatment, you can see this merge is like increasing here with the treatment of CREB. So does the CREB. The CREB is also moving to the nuclei. It's like there are more CREB get into the nucleus and FKBP51. However, after 3 days of CREB treatment, the cell is so exhausted, and you can see that the co-localization of FKBP51 to the nucleus is quite weak now. And also the CREB co-localization with the nucleus is also much weaker, suggesting exhausting condition, and this maybe is related to the stress-induced disease. And then we confirmed all the previous data with Western blot analysis. And we also treat the cells, the neurons, the cultured neurons with various concentrations of CREB, 100 nanomolar and 1 micromolar. And then we extract the nuclear fraction. Then we look at the Western blotting and to see if the nuclear fraction has increased the FKBP51 or CREB, and the total protein level is changed or not. What we found is like after 30 minutes of treatment in the nuclear fraction, the FKBP51 is significantly increased, and the CREB1 level is also significantly increased. However, the total level of FKBP51 is decreased. And so that may be that after this 30 minutes, this molecule is kind of used up a little, and the CREB concentration in the low concentration is increased, but the high concentration does not change. So that this increase is not because of the total protein change. After 24 hours treatment with quart, the neurons, we also extract nuclear fraction from the neurons, and we also see an increase of FKBP51 and CREB, which is supporting the double immunostaining data, and the total protein level is increased both in FKBP51 and CREB, suggesting that there is a compensation on the use up of these two molecules. So the 24 hours quart treatment, the cells are still holding up. After 3 days of quart treatment, you can see a significant reduction of FKBP51 and CREB in the nuclear fraction. So that you can imagine that those transcriptional regulation is not there anymore. So the protein synthesis can go down, and all other consequences of this nuclear fraction reduction. And also total FKBP51 and total CREB is also reduced. Then we want to see if this CREB localization to the nucleus depends on FKBP51 or not. Then we used this FKBP5 gene knockout to see if this gene knockout can block the translocation of CREB into the nucleus. We found that you can see the green is FKBP51 and the CREB is the red. And after this animal, wild type animals were injected with 4 grams of corticosteroid or 40 milligrams per kg of corticosteroid for 3 hours, you can see that there is a significant increase of the FKBP51 in the nucleus, and also the CREB in the nucleus. In the knockout mice, the FKBP51 is like cut off, like by that the amount is reduced by about 50–60% already, but it is still there. However, you cannot see much of the increase of the CREB. The co-localization of these two molecules are quite significantly reduced, compared to the wild type. This after 3 hours treatment with quart, the translocation is significantly reduced. Then after 7 days of treatment, you can see that the CREB nuclear translocation is quite abolished, and so that this FKBP51 is quite critical for the CREB nuclear translocation. So this knockout also FKBP51 is also not much in the nucleus as well. In the knockout mice, that this although there's still it's like a as heteros, so you can still see some FKBP51, but it's there is no significant increase. After 3 hours treatment, the CREB and the DAPI co-localization also shows similar result, that that in the wild type you can see the translocation of CREB, but in the knockout mice you cannot see much of the translocation anymore. So that suggesting that the translocation of CREB to the nucleus depends on the FKBP5 expression. So after 3 hours treatment, the nuclear expression of FKBP51 and the CREB were increased in the PFC, which is like in the wild type is increased, but in the knockout mice, there is a significant decrease of FKBP51 and CREB. So the co-localization of FKBP51 and the DAPI also is quite low in the knockout mice, especially in the hetero of FKBP51 after 40 milligrams of treatment after 7 days treatment. So that core treatment reduced the CREB localization in the cell nuclei in the PFC of FKBP knockout mice. After 7 days of treatment with CREB, you can see that the translocation of CREB is quite low compared to the controls. And we also confirmed this by Western blot analysis. And we extracted the nuclear proteins from the prefrontal cortex of this transgenic mice. And we found that the CREB, is significantly reduced in the nuclear fraction. After especially the 40 milligram after 40 milligram treatment, and the heterogeneous is the knockout mice shows even more significant reduction of FKBP51 and CREB. And next, we want to see the CREB regulated anti-inflammatory protein BDNF, TGF beta, agenus 1, and IL-10 levels. These 4 proteins are the downstream of CREB, and those proteins also are anti-inflammatory molecules, BDNF, TGF beta, arginine 1, and IL-10. Those are anti-inflammatory molecules. Those are, there is a balance of inflammatory and anti-inflammatory in those brain region. And we found that after 3 hours of treatment, in wild type, you can see an increase of the BDNF, but in the knockout mice, there is none. And for TGF beta, and in the wild type, there is an increase after the treatment for 3 hours. And you can see a reduction in the hetero knockout mice. For arginine-1, and also after treatment, there is a significant reduction. So does the IL-110, suggesting that the balance of inflammation and anti-inflammation is tilted, is much towards the inflammation side in the knockout mice. So this is after 7 days of cord treatment, and you can see that even in wild type, you can see a significant reduction of BDNF. Arginine-1 is kind of, but the total IL-10 is also significantly reduced compared to the low dose treatment. But in the knockout mice, you can see that it's always a significant reduction from compared to the hetero or the controls. So in the knockout mice, this mechanism is quite damaged. Then we look at the FKBP5, the role of FKBP51 in cord-mediated anxiety-like behavior. After treatment with 3 hours with cord in elevated plasmates, which is an anxiety model, you can see that in the wild type, there's not much change, but in the heteros, there is a significant reduction compared to the wild type. So that it just sensitized the whole thing. And also distance traveled showed not significant, but is a strong decreased trend in the number of buried marbles, which is a test for repetitive behavior and anxiety, and there's no change after 3 hours of treatment with cord. After 7 days of treatment, you can see that in the heteros, there is a significant reduce in the times in the open arms, and the distance in the open arms, there is a significant reduction. In the number of buried marbles, there's also a significant increase in the number of marbles buried, suggesting that this knockout of FKBP51 will sensitize the anxiety, the stress-induced anxiety behavior. So make a conclusion that the FKBP51 formed a complex with CREB and moved to the nucleus in a dose-dependent manner. In the process of cord response, FKBP51 is the key to mediate the regulation of CREB function in the nucleus. In response to the cord stimulation, that FKBP51 CREB is a signaling pathway regulates the anti-inflammatory factors expression like TGF beta, BDNF, IL-110, and arginine-1. And in response of cord stimulation, the inhibition of FKBP51 CREB immune repair signaling pathway will promote the occurrence of anxiety-like behavior in animals. So make a summary of the mechanism that you can see that the FKBP51 after GR cord or activate FKBP51. It forms complex and helps the CREB get into the nucleus. And this CREB will stimulation will express many molecules like BDNF, TNFR, TGF beta, IL-10, etc. And it moves the inflammatory apoptotic status towards the function, the direction of anti-inflammatory regeneration status. Therefore, it leads to the inhibition of the anxiety behavior. On the other hand, if the system is exhausted, FKBP51 is gone, then this synthesis regulation will go towards the inflammatory apoptotic status, which will lead to the anxiety-like behavior. And I would like to thank my colleague and Beijing Capital Medical University and Dr. Gang Wan, and also Lin Zhan. And in the Yunnan University, this work is mostly done by Yang Ho, a graduate student in my lab. And I will also like to thank all these foundations and all the help from the people in our group. Thank you.
Thank you so much for your presentation. Now floor is open for the questions if anyone have. If anyone having the questions after the presentation also, they can type over the chat box and the particular person will respond back. Thank you, doctor, for your wonderful presentation.